Hello everyone, thank you all for being here today. I stand before you with a deep passion to present similar or distinct, multiple ways to calculate brain age. Normative modeling of brain development has become a topic of interest for developmental cognitive neuroscience. Normative modeling explores the typical patterns of brain development across population, and it provides valuable insight into how the brain evolves and functions during different stages of development. One compelling application of normative modeling is the concept of brain age. Brain age is indicating the deviation of an individual brain maturation from the norms. To have more details, machine learning framework should learn the association between age and features. After cross-validation, trained model going to test in unseen data. The predicted age called brain age and subtracting chronological age from brain age called brain age gap that could be positive or negative. Positive brain age gap show the older brain and negative brain age gap shows the younger brain. Previous studies highlighted that brain age served as a biomarker used by researchers to assess its association with behavioral scores in individuals. However, there are inconsistencies among studies regarding the association between brain age and behavioral scores. In other words, some studies have reported no relationship between brain age gap and variable of interest, while some studies reported significant association. The utilization of different models for calculating brain age might be a reason for the inconsistencies observed in studies examining the association between brain age and variable of interest. The variability in calculate brain age explained by using of different machine learning algorithms and brain imaging modalities and different brain features. Some studies investigate that using different machine learning algorithms produce a similar brain age gap or not. These studies reported the good agreement between brain age gaps that calculated from different algorithms. However, studies rarely consider the fact that each feature of the brain captures distinct aspects of brain development, and it is suggested that the combination of different features can increase brain age accuracy. Therefore, a comparison between different measures of brain age gap is needed to see if they capture similar information and overlap. Secondly, it would be important to assess the assumption that combination of brain features increase the power to capture behavioral scores or only increase the accuracy. Therefore, the aim of this study include calculate brain age from different features of brain. Secondly, investigate the difference between the accuracy of each brain age model. Thirdly, investigate the relationship between different brain age measures. And lastly, investigate the power of each brain age measure in capturing the variance of behavioral scores. For the methods, we use the data from the HCP lifespan, include HCP development, HCP young adulthood, and HCP aging. The sample size was 2400 and the age range was 8 to 100 years old. We used T1 images and used different features such as cortical thickness, cortical area, cortical volume, subcortical volume, and combination of all cortical features and subcortical volume. Additionally, we consider vertex-wise features including myelination, curvature, so-called depth, and thickness. For the analysis, we used loss of regression with tenfold cross-validation to calculate brain age, and Pearson correlation was used to investigate the association between brain age gaps. Mixed effect model was used to assess the association between brain age gaps and different cognitive scores. For the result, I should mention that we observed cortical area has the worst accuracy in our study, and the combined measure that use all cortical features including cortical area, cortical volume, and cortical thickness plus subcortical volume has the best accuracy. In In the vertex-wise feature, I should mention that curvature has the worst accuracy and vertex-wise thickness has the best accuracy. This plot also shows the correlation between each pairs of brain age gap. Higher correlation are color red and lower correlation have paler shades. In terms of looking at the association between different brain age gap, I should mention that there was some features that have high correlation together, though most of them have a smaller correlation. For example, in the association between the brain age gap that can calculate from the combination of cortical thickness area volume and subcortical volume with the brain age gaps calculated only from each feature separately, I should report that brain age gap from cortical area has a smaller correlation with the combined brain age gap, and other have higher correlation. Regarding the report of correlation between solo brain age gap, I should mention that most of them have small correlation. This can reflect that they have unique information and capture different aspects of brain development. We also should mention that the highest correlation was between cortical thickness and vertex-wise thickness that have a similar nature. About association between different brain age gaps and cognitive scores, we use mixed effect model and keep sex and age as a fixed effect and size as a random effect. We observe that cortical area that has the worst accuracy always can capture the information from all cognitive scores. However, cortical thickness could not capture the information from the any cognitive scores, and cortical volume has no relationship with fluid cognition. Interestingly, combined measure that created from cortical area volume thickness and subcortical volume and had the best accuracy always have a less power to capture cognitive scores, and there was no relationship between combined brain age and crystal cognition. About vertex-wise feature, I should mention that although thickness and so-called depth had a very high accuracy, they could not capture any cognitive scores variance. However, curvature always has a good power to capture the cognitive scores variance. In summary, about the accuracy, our results showed that the combination of the cortical subcortical brain age as well as vertex-wise thickness brain age has the best accuracy, and the cortical area and vertex-wise curvature has the worst accuracy. Regarding collinearity, our results showed that there is a varying degree of association between different brain age gaps calculated from different features. While some features are more similar, up to 0.7, most measures of brain age capture different developmental trajectories, less than 0.3. And about cognitive scores, different brain age models have different power to capture cognitive scores. The brain age model with higher accuracy are not necessarily better to capture cognitive variation, and combining features increase age prediction accuracy but had detrimental effect on detecting cognitive variation. In conclusion, the higher accuracy cannot guarantee the power of brain age model in terms of capturing the variable of interest. Combination of different models may not add to capturing variable of interest, it can even mask the neurological mechanism behind behavioral variations. And also, it would be important to distinguish between different measures of brain age computed from different brain features. At the end, brain aging can be assessed using different normative clocks. These clocks may be related to different underlying process and different mechanisms and should hence not be combined without appropriate consideration. For future step of this study, I want to mention that we have planned to use different modalities of brain such as functional and structural connectome as well as FA and MD, and comparison between common brain atlas that used in the field to calculate brain age as well as high resolution features. I should thanks to my wonderful supervisory team and my colleagues who helped me to conduct this study, and thank you for listening to me.